purpose of this experiment is first to perform hemoglobin estimation using Sirleaf's acid hematine method. The second purpose is to determine hemoglobin concentration in a given sample via Sirleaf's acid hematine method.
I will continue with listing down all the equipment and materials needed for this experiment. The equipment needed to run this experiment includes hemoglobin pipette or hemometer pipette, comparator box with a brown glass standard size graduated hemoglobin diluting tube or hemometer tube, glass rod, proper 19% ethyl alcohol, sterile cotton and a needle. As for the materials, distilled water and N-proton hypochloric acid.
Next, I will continue with the procedures for this experiment. The first step for this experiment, N10 of hydrochloric acid was added to the hemometer tube up to 20 mm cube mark. Then, the hemometer was gently placed in the stand at the comparator box. Next, the tip of one finger was sterilized with 90% ethyl alcohol. The finger was cleaned in order to prick and draw blood using a sterilized needle. 20 microliters of blood were drawn into the hemoglobin pipette. While doing so, make sure to avoid air bubbles moving into the tube. Then, the blood was immediately transferred into the hemometer tube and mixed properly with hydrochloric acid. The mixture was then left in stand at the comparator box at room temperature for about 10 minutes to allow conversion of hemoglobin into acid hematine.
After 10 minutes, distilled water was added drop by drop until the color of the mixture matches the brown glass standard. The brown glass standard represented 100% hemoglobin with reference to a standard which was 14.8 gram% of hemoglobin per 100 ml of blood. When the color has matched, the hemoglobin concentration was read directly to the hemometer tube by noting the lower meniscus of the diluted mixture. The reading was then being recorded for the tabulation of results.
The result from this experiment is the normal value of hemoglobin level in human are different between male and female. For adult male, the normal value range from 13.5 to 70.5 gram per deciliter, whereas for adult female, the normal value range from 12.5 to 50.5 gram per deciliter.